Miss Danny? Miss Danny, you're on mute again.
How you doing?
I'm doing good. I finally caught it.
Oh no, when'd you catch it?
We're thinking Thursday.
I'm sorry. That's awful.
Nah.
Yeah,
Can't
right.
breathe through my nose. Augie, stop that. I brought Augie in with me so everybody could see. Say hi, Augie.
Augie, how are you, buddy? I've got allergies that are making. Hey, buddy. I've got allergies that make my voice sound terrible, but it's just terrible allergies. This one is better.
I hate allergies.
Yeah,
They suck.
yeah, they do. They're terrible.
I can see you. That one works.
Now you can probably see more of the room because that's a wide angle. So, what you doing, Og?
Say, I'm starting potty training.
Oh, hooray!
Yeah.
Won't it
And
be
I
the
pee in
day?
my underwear every day.
I was gonna say, won't it be the day when there are no more diapers in the plaster house? Woo!
Oh, wow.
Y'all have kept the diaper companies in. What's up, buddy? How you doing, Squish?
Can you say hi? Hi. Say hi.
He's so funny.
He's very attached to this flute right now.
Recorder.
Okay.
Awful sounding things.
Yeah. One night, Augie had that in his bed, and he just blew it all night long. Like, he kept it inside of his mouth while he was sleeping.
No.
And so, it was like.
That's all the nose. All the nose in Nopeville right there. That would make me crazy.
I finally got down there and took it out of his mouth, and when I did, he just like got rid.
Oh my goodness. Hey, give me a sec. I gotta go deal with Ozzy while people are coming in. So just a sec. Hey, come in. Have a seat. Ozzy.
It's Danny.
So my voice is scratchy because I've got allergies, but it really is just allergies. It's just it sounds terrible. I don't feel terrible. I just sound off. You can put it right back in your binder. I got it. I will mark it for you. I'm gonna make myself a note over here. Lily, binder work. Hello, how are you? Good to see you guys today. We're excited. I have to put you in our Facebook group. I've realized I forgot to do that last week, but we do have a group where moms can chat and stuff. And sometimes I'll send messages over there that are not, like everything will come through email that's official, but I'll put stuff over there too.
Okay.
Yeah,
Okay, sounds good.
great. Yeah.
So he bought this
And
binder, but
he's got one right here. I was about to say, Anthony, look on the dining room table. There is a binder just for you. It's all yours and it's all set and put together. So you can do whatever you want with that one or whatever, but it's right here. It's got everything all put together for you. So last week's lab is going to take 2 weeks to complete. So today you're going to look and see where everything is, kind of get some idea. And then next week you're going to look one more time. And then after that, you will keep it in your binder, but you're going to need to write on it for the next couple weeks. So
yeah, because me and Savannah were like, is this keeping binder?
what am I doing with this? Yeah.
I was like, no, I think this Danny said it was like a 2-week thing.
It's a 2-week thing. Yeah. It was in the email. The email said so. All right. So Drew's still coming down. All right. Darcy and Lewis are in South Carolina on vacation. Thanks, Nakia. Go have a nice time by yourself. Enjoy this little respite every week. Make it your, I don't know, shopping time if that doesn't stress you out.
Yeah.
If it does stress you out, do something else.
Dog off the daycare and I'll figure
There
out
you
something.
go. That's great. All right, cool.
Okay.
Thanks. All right, so I've got Claire up here somewhere. She's in here and let's see. So like I was saying, my voice is super duper scratchy because I got allergies. Your Q2 binder contents, let me grab them for you. They are right here. Do you have your binder in your bag? All right, let's set this up real quick and then we can start our list. All right, so let's see. I'll let you put these two back where they go. Okay, so what we're gonna do is we're gonna take out your syllabus. We're gonna take out lessons 8 through 17. Because now they're updated. Ditch. So be sure you keep your binders organized, and you guys are doing a good job, but when you get to studying for your midterms and stuff, you're gonna wanna know where to find stuff. And it'll be terrible if you can't find it, you're studying for it. Yoke. So hopefully I won't get a weird coughing fit, but along with this like strange scratchiness I get, I have this weird one little tickle spot that keeps popping up in my throat, and then I cough like I have COVID and I'm dying, but it's neither of those things. So let's do our lesson. Is everybody ready? Are we good to go? I don't know where my son is, but he didn't come down and get his stuff either. Well, okay. So we're starting our new unit. Unit 4 is going to be on genetics. So this should be a lot of fun. I really, really, really love this topic. It's really, really fascinating. And today's lab is going to be kind of a mixture of last week's, we're going to catch up on data from last week, and then we're going to do some modeling. Drew, if you could take a minute to take your stuff somewhere and then come sit down. Okay, thank you. Anybody been to the fair yet?
No. I want to go so bad though.
Did you go? Yeah.
Michael went last night with a lot of friends from church.
Yeah, I heard he did.
I love the fair, but I can't go.
I know, I'm sorry. It's no fun. So Drew and I were at the fair on Sunday night, and we're going to be there again tonight because Drew is with the Civil Air Patrol, and they have a booth. So we've been up there at the booth doing stuff. Yes, modeling your jacket. All right, go put it up. Put it somewhere. I don't care where. Put it in the guest room on the bed. Just close the door. All right, so genetics. We're going to do genetics now. And this should be fun. This should be a really fun topic. So this dude, Mendel. Did we read about Mendel at all? A little bit? All right, I love this guy. He's definitely one of my favorites. So if you guys remember, we've talked before, a lot of scientists way back that made some of the earliest discoveries, a lot of them were either like really wealthy people or religious people. And that's because they had time on their hands. So a lot of the really wealthy people, like independently wealthy people, they could just spend their money on whatever they wanted and do experiments and buy equipment and have a lab and whatever, just because they felt like it. They didn't even really have to have a formal education in science. Well, Mendel was a monk, an Augustinian actually, monk. Yay for Augie, right? And so he spent a lot of time in the garden, and he made most of his discoveries there working with peas. So we're going to talk about that today. So here's our buddy Gregor Mendel. So this wasn't so long ago because we have a photograph, right? So he was born with a different name, and when he became a monk, they changed it. So he's Gregor Johann Mendel, 20th of July, 1822 to 1884. So that's a pretty good lifespan for somebody living in the 1880s, 1850s, 1860s. So he was a polymath. Anybody remember what a polymath is? It's a great word. It's a lyric in Hamilton. I always remind y'all, it's a lyric in Hamilton. Yeah, a polymath is somebody who is well educated in a wide variety of topics that don't necessarily have anything to do with it. Yeah, Hamilton refers to himself as a polymath in one of the songs. Another great.
So like he thinks he's amazing.
Yeah, another good example of a polymath, like probably one of the best known American polymaths, would be Franklin or Jefferson. They were all polymaths. So Mendel was a polymath. He was a meteorologist, a mathematician, a biologist, an Augustinian friar, and the abbot of St. Thomas Abbey in Brno, Moravia. So interestingly, that little town there, Brno, see the name of this town? It's funny to me because several years ago, Jason and I visited the Czech Republic, and then we drove to Hungary, and we had to drive through Slovakia. Well, that used to be Moravia, and this town is still there, and I didn't realize it at the time, because if I had, I would have gotten out and gone and looked, but I would have gone and found all his stuff, but I didn't realize it at the time. But there's no vowel in those first three letters, and it became a thing that we joked about for the entire drive, because in English, you have to put a vowel somewhere. It's either Brno or Borno, or it's Brno, or where's the vowel go, before or after the R? I mean, it has to be a vowel, right? But I asked a friend whose husband is Czech how they actually say it, and it's Brno, kind of like Bruno, sort of. So anyway, Johann Mendel was born the son of a struggling farmer and in a German-speaking family in what today is the Czech Republic. And he was given the name Gregor when he joined the Augustinian monks. He became a monk in part because it enabled him to obtain an education without having to pay for it himself. He didn't have any money. So if he was going to get an education, the only way to do it was through the church at that point. So in the monastic life, in his words, he said it spared him the perpetual anxiety about a means of livelihood. That's a great quote. So being a monk means that it spared him the perpetual anxiety, or like the ongoing stress, right, about a means of livelihood, making a living. Took away the stress of working, basically. So he gained posthumous recognition. What's posthumous? You know what that word means? So posthumous means after you died. So this means that all of the credit that he gets for being what we call the father of genetics came after he'd already died. He didn't get that recognition in his lifetime. So posthumous recognition as the founder of modern science of genetics, and sorry, and his pea plant experiments conducted between 1856 and 1863 established many of the rules of heredity, now referred to as the laws of Mendelian inheritance. So he coined the terms we use today, recessive and dominant, in reference to certain traits. And he published his work in 1866, demonstrating the actions of invisible factors that we call genes now, in predictably determining the traits of an organism. So the profound significance of his work, like I said, wasn't recognized until after he was gone, around the turn of the 20th century. So 30 years after he was dead, they were like, oh, dude, this guy was onto something, right? And it's his laws and it's his concepts that give us modern day genetics. So just for background, this is a diagram of a pea flower. Okay, so a pea flower, this little thing at the bottom, this is like what a bud would look like if it was closed and then it opened up. When it opens up like that, it's called the sepal. Sometimes you'll see parts similar to this labeled calyx. It's the same thing. And then this thing right here, this whole structure together is the pistil. That's from here to here. And this is the female part of the flower. So this is the part that produces the ovule, or we call them ova when we're talking about mammals, but basically the flower eggs, if you were to make a parallel term for mammals, right? And then this is the ovary, so that's where they're produced. And then this right here is a big long channel that leads down to where the ovum are kept, or the ovules are kept. Now, here's an interesting thing. This right here, if this was an apple blossom, this would be what becomes the apple. So when you eat an apple and you have seeds inside, right, it's because you're eating this big part here. And that makes sense because it gives the seeds a nice nutrient medium to grow in, right? If the apple just falls on the ground, they have all the nutrients from the apple, plus it makes you eat it. And if you're a chipmunk and you go and poop the seeds elsewhere, you're helping the apple tree spread. So with peas, they're just set up a little differently and they make peas inside of a pot. Now this right here, the stamen, this is the male portion of the flower. So this is the anther and this is the filament. The anther is this little thing on top. You ever seen those in flowers? This little guy right here? Okay, that's where the pollen is. The pollen is the male gamete. That's the male sex cell of a flower. So when you look at flowers, you can't see the female sex cells because they're inside of the ovary. They're inside of the flower. But the male sex cells are the pollen that you see. So in the spring, when there's pollen everywhere, it's tree sperm. It totally is. It's male tree sex cells everywhere, right? That's what they are. So now that you have an idea of what that looks like, that should help you explain a little bit. And you know, this is the symbol for female and the symbol for male, right? All right, so fertilization produces new cells from male and female gametes, or sex cells. We use the term gametes usually. In flowering plants, there are two types of fertilization. So one of them is self-pollination and the other is cross-pollination. So in self-pollination, flowers can actually pollinate themselves. So the gamete, the male gamete here, the pollen, touches the top of the pistil here, makes its way down to the ovary, fertilizes those ovules, and you have a new flower, but it's genetically identical to what you started with, right? Because it's using the same DNA. Cross-pollination is when you have one set of male DNA from one flower and it gets transferred to another. So bees do this naturally. So do butterflies, hummingbirds, right? What Mendel did was he went in and carefully removed these stamen and acted like a bee basically. But instead of just carrying it, bees carry the pollen on their legs, right? They have little baskets on their legs almost that catch the pollen. He would take the whole little anther with the filament and touch it to the next flower. So pea plants are usually self-pollinating. All it takes is a little wind or action and they'll bump themselves and pollinate, right? So pollen from a flower lands on the pistil of the same flower, just like that. But what Mendel did is he created a method by which he cross-pollinated or crossed pea plants. So he would breed different pea plants to one another. So he would go in here and he would he would cut out the anthers, so there's no way this little flower can pollinate itself. And then he would either take the whole anther and filament, or he would take a brush and collect the pollen and then stick it on the pistil of a neighboring flower, and that would cross-fertilize or crossbreed the pea plants. So when you cross pea plants, you have what you call a purebred organism. So that's an organism of many generations of the same trait. So that would be what happens here when it pollinates itself, right, before you start messing with it. But Mendel started with these purebred plants. So he has plants that, like, every time he plants them, they give him a purple flower. Every time, always purple. Generation after generation after generation. So we're going to call that purebred, okay? And this is how he got his nice purebred line started, and he studied these 7 traits. So he studied seed form, whether it was what he'd call form or cotyledon, round or yellow, wrinkled or green. These were the options, right? Or it would be the flowers where round or wrinkled. I did it the wrong way. Round or wrinkled, yellow or green. Whether it was white or purple, whether the pod was big, full, or if it was wrinkly and constricted, whether it was green or yellow, and axial pods, which is kind of where they're positioned. See right here, these are axial pods and terminal pods. So this would mean that it's down, if you look on, like on the vine, as opposed to at the end of the vine. So at the end of the vine, it was terminal because it's at the end, and axial was in the middle. And then size, were they tall or short plants? So this little video, what do you think? This does a great background here on how he did it with graphics that move, which I can't do, graphics that move. And I could watch TED Eds all day, right? They're great.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century, the man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. One of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations.
Look how they
of genes.
squat.
Depending on which type of allele Mendel found in the
See?
seed, we can have what we call a homozygous seed, where both alleles are identical, and what we call a heterozygous seed, where the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers lowercase friend, so the only time you get green babies is if you have 2 lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the 2 heterozygous kids marry, their babies could have any of the 3 possible genotypes, showing the 2 possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
So Punnett squares are actually kind of fun. They're like logic problems, and we're actually going to do some when we get through this stuff. So Mendel had his law of dominance. All right, and what that means here is that genes are going to pass their characteristics or traits, we're going to call them traits, from a parent to an offspring, and that different forms of a trait are called alleles. So when we're talking about genes, we're talking about what designates a trait, we refer to it as an allele, all right? And then you have phenotype and genotype. So what's the difference between phenotype and genotype? Who remembers? They just said it and it was in your notes. Genotype is what the alleles are. So it's, in this case, AA, right? And your phenotype is what it presents as, or how it's expressed is the way we use it, what it looks like. All right. So the most basic form of Mendelian genetics, each trait is derived from a pair of alleles. Now it does get more complicated, but in our basic beginning, we'll learn about a pair. So each allele is either dominant or recessive. So dominant alleles cover, we use the term cover, the expression of recessive alleles. So the dominant allele is expressed. So if you have brown eyes and brown eyes are dominant, and you get a brown allele from your dad and a blue allele from your mom, you're going to show brown, even though you have that gene. Now, if you have a kid with somebody who has blue eyes, there is a chance that your child will have blue eyes, even though you don't, because you carry that allele and it's just covered by the brown that you have. And we're going to do a lot of stuff in the next couple of weeks that'll make all of that really clear how it works. So the combination of alleles is the genotype, and how it is expressed is called phenotype. So dominant alleles are always designated with a capital letter, and recessive alleles are designated with a lowercase letter. That's how we tell what's what. So the principle of dominance that Mendel came up with says that dominance always exhibits that form of the trait. So if you have a dominant allele and a recessive allele, you're always going to show a dominant phenotype. And if you have two dominant alleles, you're always going to show a dominant phenotype. And the recessive exhibit that form of the trait only when the dominant is not present. So in this case, when you have two recessive alleles. So when you look at somebody like me, who's like got really fair skin and blue eyes, I'm full of recessive genes, basically. I'm like very recessive, very, very recessive. So the principle of dominance predicts that each phenotype is determined by two alleles, one of which is expressed when present, known as the dominant allele, and the recessive one. And recessive phenotypes are expressed only in homozygous recessive. So homozygous recessive, we're going to talk about that in just a second here too, what that means. So the principle of segregation, this is another one of Mendel's rules. So Mendel said that the segregation predicts that during gamete production, that's the production of sex cells, each gamete receives exactly one of the two alleles present in the parental cell at random. So it's random whether a particular gamete has a dominant or recessive allele just based on what the parent cell had. That's how we get an even distribution of stuff. And then lastly, he has the principle of independent assortment. And what that means is that independent assortment predicts alleles coding for different phenotypes are segregated independently from one another during gamete formation. So this means that if you're looking at, for example, flower color, seed color, where the flowers are positioned on the vine, those things are all independent of one another. And that's true for a lot of things. Now, there's some things where they are linked, and that's when you get into traits that are determined by multiple alleles, which we'll get to. So here's probability and heredity. Here's where you get to do Punnett square stuff, and it's fun. But for a minute, we would talk about chromosomes, because we haven't talked a lot about chromosomes, and I want to make sure you have an idea of how they function and how they're structured, because that'll help you understand all the rest of this stuff about alleles. So here we go. Here's a little thing. So chromosomes exist in pairs. Can you match the pairs? Look up there with your eyes and see if you can find a pair for everything. Yep. Have you found every pair? Okay, so chromosomes exist in pairs. So we're going to match these pairs. Oh, why don't you just go? You're supposed to just go.
Oh, here we go. So there's this sandal, right? And those sandals and these guys. And then lastly, those guys there. All right, now they're called homologous pairs. Here's why that matters. So are these two shoes identical? Who says they are and who says they aren't? Who says they're identical? Are they identical? Okay. Are they not identical? Well, let me ask you this. Could you put, which shoe is this, the right or the left? Could you put it on your right foot? Ah, so they're not identical, but they go together, right? They're homologous. All right, that's how chromosomes work. It's exactly how chromosomes work. So chromosomes exist in pairs, and we call them homologous pairs. And that means that they go together, but they aren't identical. So look at your shoes and think about how they could compare to homologous chromosomes. They clearly go together, right? If you put a sandal on one foot and a sneaker on the other, that's not going to work. They don't go together. So we call those sister chromatids. They're similar, but they're not identical, kind of like your left and right shoe. So if you look at all the shoes in the classroom, okay, I've got all these shoes in the classroom here. There are no two identical pairs. So this would be kind of like all the chromosomes in one of your cells. They all go together, but none of them go with any other ones, right? So all the shoes in the class are like unique pairs of homologous chromosomes in cells. So here's a fun one. All right, these are going to be our chromosomes. We're going to match them. So what do we see? See if you can find the homologous pairs. And look with your eyes and see if you can figure them all out. You got them? All right, so if we put them where they need to go, you'll see that what you get here is the same kind of thing. Obviously, this is not the same as this, but they go together. And that's how chromosomes look. They look just like that when they're paired. So they match, but they aren't identical, just like shoes. So if you think about them like a pair of shoes, you're good. This is because one chromatid, or in this case, a shoe, right, comes from a male parent, and the other chromatid comes from a female parent. So you have the exact same number of chromatids from your dad that you got from your mom. And each one of them has a pair. So when they come together, they find their match. So here's a homologous pair, and here's a homologous pair. And this shows you that, like, the kind of indigo one here, this would be your father's copy in this case, and the maternal copy is going to be the kind of violet one. So they go together, right? So alleles versus genes. All right, so each chromatid has a series of alleles, and its sister chromatid possesses the same series, so that the chromosome is composed of alleles in pairs. It's pairs within pairs. So we have this pair, right, that we're talking about. We have like one from mom, one from dad, right? But on them are these pairs within pairs. So this pair right here, we would call these alleles. This is an allele for blue eyes. It's a little b because it's recessive. And this is an allele for brown eyes. It's a big B because it's dominant. But when we put them together, that's a gene. So you have a chromatid from mom, a chromatid from dad. They're homologous. They match, but they aren't identical. When they get together, they each have an allele that aligns with an allele on the other chromatid. And when you put the two alleles together, it's a gene. So would this person have blue eyes or brown eyes? Yeah, brown. Why? Because brown is dominant, and you know, because it's capital letter, right? So, I mean, you probably know brown is dominant because there's way more brown eyes in the world than blue eyes. But if you didn't know, you would just look at how it's labeled. So alleles versus genes, again, make sure that we're clear on it. So this right here, these are alleles on sister chromatids, and two alleles form the gene, which then defines what we call a monogenic trait. That would be a trait that is defined by one gene. Now, there are plenty of traits that are defined by multiple genes, but we'll talk just at the beginning here about what we call monogenic, one gene. There are some things that are going to be controlled by lots of genes, like I said, like height, things like hair color, skin tone, even eye color to some degree can be controlled by more than one, because it could be a co-dominance thing. Like, that's more like different alleles. But widow's peaks. So if you look at your hairline, does your hairline come to a point or is it round? Everybody think about your own. Does it come to a point or is it round? Pull your hair out. Let Lily look at you. Just all the way back. Pull your hand back. So does it come to a point? Sort of a little bit. How about you, Colson? I can't see your hair under there either. It's kind of straight. Yeah. How about you? Yours is kind of straight too, isn't it? Okay. So a widow's peak, when it comes down to a point, that is a monogenic trait. It's one gene. So this is actually a picture of Mendel's research garden for real, like back in the day when he was using it. And this is what it looks like today. They still celebrate him there today. Well, I say they now celebrate him because they didn't celebrate him then, right? So for example, the trait for height for these pea plants is represented as TT. Each letter represents an allele. This one's going to represent what? Tall or short? Tall. And how about the lowercase t? Short, right? So here's where you have to memorize some new words here. So if your genotype is tall, then it's called homozygous dominant. Okay, here's how we get the word homozygous, and this might help you remember it. Zygous comes from the term zygote, which is a single cell that has just been created from two gametes. So a male sex cell, a female sex cell come together, there's fertilization, you have one cell, it's called a zygote, okay? That's where that part's coming from. And do you remember homo and hetero mean, like homo means same and hetero means different. Yeah, different. Okay. So this is the same in the zygote for dominant, right? If you read it that way, same in the zygote for dominant. So when it's fertilized, you get dominant and your phenotype is going to be tall. This one is going to be homozygous recessive because they're both lowercase, right? So the same in the zygote recessive, going to be short. Now this one is going to be heterozygous because look, they're different. And that implies dominant. You can say heterozygous dominant, but it implies that it's dominant. You can never say heterozygous recessive. Doesn't work. Why? Why can't you say heterozygous recessive? Why is it not a thing?
Because you have two lowercase.
No, that would be here. That would be homozygous recessive.
Different lowercase.
If you were saying heterozygous recessive, you would be saying that this capital T and this lowercase t are going to present with the dominant trait. And going to present with the recessive trait. And that never happens because the dominant trait is always going to cover the recessive trait. So, and we'd also could call that a hybrid. All right, so you have these Punnett squares in your notes. So it says, let's say in seals that the gene for length of whiskers has two alleles. The dominant allele is a capital W for long whiskers, and the recessive allele is a lowercase w for short whiskers. So what's the probability of producing offspring that have short whiskers from a cross of two long whiskered seals? So two long whiskered seals would present like this if one is homozygous dominant and one is heterozygous. Okay, so tell me, is the homozygous one written vertically or horizontally? Homozygous means the same, the allele's the same. So is that the one written vertically or horizontally? Which one's homozygous? This one or this one? This one's horizontal and this one's vertical. So this one's homozygous because they're both capital. And this one's heterozygous because you have a capital and a lowercase. So when you cross them, you get a W from here and a W from here that are both capital. A W from here and W from here that are both capital. And this one, you're going to get a W from here that's capital and a W from here that's lowercase. So how many of your seal offspring are going to have long whiskers? What percentage? Think about it. Look at it. What percentage are going to have long whiskers? You probably answered. What do you know?
50.
Oh, 50? You say 50? It's going to be 100. And here's why. Because capital, 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 capital. These are all dominant, dominant. Dominant recessive is going to show dominant. Dominant recessive is going to show dominant. So they're all going to have long whiskers. All right. So what about this one? If one parent seal is heterozygous long whisker, and the other is short whiskered. So that would look like this. Here is your heterozygous long whiskered, got a dominant allele and a recessive. And here is your short whiskered. The only way to be short whiskered is to have 2 recessives. So what do we get here? Boom, 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 boom. All right, what percentage of your seal offspring are going to have long whiskers? 50, right? And what percentage are going to have short? 50. Good job, Sky. So in purple people eaters, 1 horn, which we're going to call dominant with a capital H, is dominant, and no horns is recessive. So let's see if we cross 2 hybrid. So that would mean they have both a dominant and recessive allele. So both these parents have horns that you can see, right? Because they have dominant allele. So let's cross them, and what do we get? So how many of each type of offspring are we going to get? Somebody tell me. How many horns and how many with no horns? 3 are going to have horns. That's right. And then 1 is going to have no horns. And that's this one here. Very good. All right. So looking at this, which was in your homework, I think, right? All right. Looking at this, and the homework was for turning in person. So be sure you put it in the folder up there. Yeah, look at yours. All right. So what's the name of Irene and Leo's sons? And what's the name of their sons-in-law? So who are their sons? Somebody tell me. Gordon and Tim and Richard. Good. Oh, I missed. No, Tim's not their son. Tim is their son-in-law. Cheryl's their daughter. So Richard and no, wait. Oh, sons, and then their son-in-law is Tim. Yeah. Oh, I didn't write Tim. Sorry, I should have. How many grandchildren? Let me fix that before I forget it. I should have written Tim there. Poor Tim. So Tim is their son-in-law. I'll put him separate. Come on. All right, there we go. So, all right, so how many grandchildren do Irene and Leo have? And how many of their grandchildren are girls? So here are their grandchildren. 1, 2, 3, 4, 5, 6. And how many of them are girls? 3. And you know, because of the circle. So if they had a name like Taylor, or I don't know, what's another good unisex name? Alex, great examples. You could always look at whether it's a circle or a square. Jesse, yeah. Usually that one's spelled differently, but not always. So what's the name of Ralph's father? Very good. And how about Ashley's mother? Perfect. What's the name of Emily's son? And how about Tim's son? Right. So after this pedigree was made, Richard and Emily had another son who they named Roger. So where would Roger go? He'd go right there next to Richard. And then Juliet married a man named Robert. Where's Juliet? Here she is. She marries a man named Robert, and they have a daughter named Elizabeth. So there would be Robert, and I didn't have room for Elizabeth, but she would be right here. And then Zach married a woman named Jean, there we go, and had a son named Craig. So that would be what it would look like if you added all of them. All right, and I'll leave that up for a second if you want to check it real quick. And then we're gonna do an activity in the last 5 minutes, or we'll start it. No, you're good. I see it.
Thank you.
I see it. You're good. And this will be, obviously, you can go back and check it. All right, so here's an activity. We're going to breed
Miss
dragons.
Danny,
Yes, ma'am.
how am I supposed to turn in my homework?
You can send it as a PDF.
Okay.
All right, so here's what we're going to do. I'm going to divide you into 2 teams. We're going to have a girl team and a
I
boy.
love dragons.
Yay, we're going to breed one. You kind of just get to watch since you're not. Actually, I can put you over there with the girls. So here's what we're going to do. We have about 5 minutes or 6 minutes. We're going to start this. We may not finish it. But right here is going to be our alleles for dragon phenotypes. All right. So what you're going to do is you are going to, on your teams, create a dragon. Okay. Put its name at the top. And then decide what its genotype is. Be sure to write what its phenotype is. So where it says on here allele. So you're going to put like whether it has 2 big B's or a big B and a little b. You see what I mean? So like for nose, under B, does it have 2 dominant alleles, a dominant and a recessive, or 2 recessive alleles? Okay, you're going to write that down. And then you're going to write its phenotype over here. All right, we're going to start this today. And then you guys are going to have some time to come up with your dragon, basically design your dragon, right? And then we'll make them later and see what kind of babies we get. So Claire, I'm going to move you over there in just a second, but let me give this to them first. All right, so one's y'all's. This one's y'all's. Three, you might want to come over here where everybody can get together for a minute. And Claire, I'm going to scoot you over to them. They can see you. All right, ladies. Now y'all can see that up there, right? I'm going to put Claire in this space here. Whoops. You can actually see it a little better. Okay, there we go. All right, so go ahead and start designing. Drew, go there. Then start designing. Pick your characteristics out. And I'm going to hand you your progress reports. I did mail them, but email them, but this is paper ones. So you can put it in your binder. Okay, so start picking, girls. And Anthony, you don't get from all this because you haven't been here long enough to worry about it. Get you there next time. Colson, that's yours.
Right. Spiky nose or no spiky nose?
I already gave you those.
Spiky nose. Where do we write it down?
Right there on that paper. So where it says, see where it says alleles?
Yeah. Do we just write down the big B or the little b for it?
Yeah, so for example, here, let me just show you what the next slide looked like. So if you were going to do, if you're mama dragon, and we'll do this, this is when we put them together. So we're not putting them together yet. So actually just do like, if it's big B, little b, or is it little b, or is it big B, however you want to do it.
All right.
Okay. And then in the next square that says genotype, you just squish them together.
All right.
Right. And then phenotype, you write out how it's going to look. Okay, so Drew, y'all should be doing that. All right, so she can help y'all decide.
Wings or no wings?
Probably no.
No wings. Well, we could do big D, little d. See how that comes out.
So first of all, write all those down.
No, you don't have to write this down. The first one is nose spike, right?
Yeah.
So you're going to decide whether it's big B, little b, little b, little b, big B, big B.
Fun
You're
that.
going to write it down
So
where
we
it says
just, we
alleles.
just
Fun
put
that.
down what we
Yeah,
want.
put down whatever you want.
Okay.
What do you guys think?
First, and this is first and second, that's dominant and recessive.
No, it's just 2 different alleles because 2 alleles make a gene. You can, they can both be dominant, they can both be recessive.
Do you
Right.
want to do little g, little g?
Okay, so
For the horns?
first of all, first one I want is not on
Or do you want to do big D?
If
big
you want
D?
it to have a nose spike, it can be one
Or
of
we
2
could
things, right? It can
do
be
little
big
g,
B, or
big
it can
G?
be big B, little B.
Yeah, probably.
You
Big
decide.
D, little
Big
g?
B, big
All right.
B, and big
Okay.
B, little B. Yeah.
So you put 1 in each block, right?
Would be
And then 1
Back
here.
spikes, no
Big
back
B, big.
spikes.
Just one letter in each of these 2.
No
So
back
is it gonna be
spikes?
big
So little h, little h.
Yeah.
big D, little B, little B.
So what, you only get 1 set. So is it gonna be
Long tail, short tail.
homozygous or
We could do
heterozygous?
big eye, little eye.
Yeah.
No, you're just making 1. Later, he's gonna get to be a dad. But right now, he's just 1. So
Red
like
eyes or
put a
yellow
big
eyes?
B or a little B
Big
here,
A.
one of these.
Big eye.
Okay, and then in the next space here, you write big B, because that's what you just created.
Okay, so big B.
Yeah.
Blue
And
spots
then
on
the
neck or
next
no
square, you
spots on neck?
write phenotype. So what does that mean? Big B means what?
Yes.
No
Yeah,
spike.
do you want
So
to do
write
big
no spike.
L, little L?
Yeah, probably.
All right. Wings, I always wings.
And you give you guys about 1 minute and then.
Tail spikes or no tail spikes?
So give it
I don't know.
a
Probably
biggie,
big
a
and
little
big.
bee.
Sure.
So big bee. Little bee. These,
Fangs or no fangs? Big and little one?
Yeah.
heck 3 times longer than head. No.
Green body or purple body?
Yeah.
Come on in. All right, so we'll do more of it next week. It's gonna take us a couple weeks.
No, we were so
So-
close.
Put
We literally
your
did
names,
this
if you haven't named the dragon yet, then just put your names on the top and hand it back. And I will,
What do you want to name again?
Sky Lily and then Claire.
Talk
Should we
to
write
Magic
Claire?
I say Coffee Magic again. The reason I say Coffee Magic Dragon is because that was the
Uh-huh.
original.
All right,
one
Claire,
for
I'm
the
sorry.
Secret 30.
All right, let me borrow your pen.
The original
Can
one-
you guys put that on like the forum because I'm really good at drawing dragons. So
Oh,
once you
cool.
come, I could draw it for us.
Okay. Cool.
How much for names?
All right, we will do that. I'm gonna let you go, Claire. We'll talk to you later.
Coffee Magic Dragon.
Bye, honey.
Coffee Magic Dragon.
Let's see.